Morning, everyone. Welcome to Temple Neurology Neurosurgery Grand Rounds. This is the last grand round for this academic year. And we have invited Dr. Miguel Nicolelis. He is logged in from Brazil. And we highly appreciate you doing this grand round for us, sir. So I'm going to introduce Dr. Nicolelis. It's a daunting task to introduce an authority figure like him, but I'll try. So Dr. Nicolelis is MD, PhD, is a distinguished professor emeritus of neurobiology at Duke University, where he was the Duke School of Medicine Distinguished Professor of Neuroscience and the founder of Duke's Center for Neuroimaging. He is founder and scientific director of the Edmund and Lily Safra International Institute for Neuroscience of Natal, Brazil. Dr. Nicolelis is also founder of the Walk Again project, an international consortium of scientists and engineers dedicated to the development of an exoskeleton device to assist severely paralyzed patients in regaining full body mobility. Dr. Nicolelis has dedicated his career to investigating how the brains of freely behaving animals encode sensory and motor information. And as a result of his studies, Dr. Nicolelis was first scientists to propose and demonstrate that animals and human subjects can utilize their electrical brain activity to directly control neuroprosthetic devices via a brain-machine interface. For the past 30 years, Dr. Nicolelis pioneered and perfected the development of a new neurophysiological method known today as the chronic multi-site, multi-electro recordings. Using this approach in a variety of animal species, as well as intra-op procedures in human patients, Dr. Nicolelis launched a new field of investigation which aims at measuring the concurrent activity and interactions of large populations of single neurons throughout the brain. Through his work, Dr. Nicolelis has discovered a series of key physiological principles that govern the operation of mammalian brain circuits. Dr. Nicolelis' pioneering BMI brain-machine interface studies have become extremely influential since they offer potential new therapies for patients suffering from severe paralysis, Parkinson's disease, and epilepsy. And today, numerous neuroscience laboratories in the US, Europe, Asia, and Latin America have incorporated Dr. Nicolelis' experimental paradigm to study a variety of mammalian neuronal circuits systems. His research has influenced basic and applied research in computer science, robotics, and biomedical engineering. Dr. Nicolelis is a member of the French and Brazilian Academies of Science and has authored over 200 manuscripts, edited numerous books and special journal publications, and holds at least 3 US patents. He is the author of Beyond Boundaries, the new neuroscience of connecting brains with machines and how it will change our lives, the relativistic brain, how it works and why it cannot be simulated by a Turing machine, and finally, the true creator of everything, how the human brain shaped the universe as we know it. So without further ado, let's welcome Dr. Nicolelis.
Well, thank you very much. Thank you for the kind introduction. It's a great pleasure for me to be able to talk to you guys. Sorry, I have been in Brazil. I wish I could be there because you may not know, but about 35 years ago, I was a postdoc in Philadelphia at the Hahnemann University, and I used to visit Temple with my then mentor, John Chapin, and we had a nice visit, several visits, and a small collaboration at that time. So I consider myself an adopted citizen of Philadelphia, and to this day, I still suffer with the Eagles and the Phillies. So it's a great pleasure to be here. I'm going to share my screen. If you just could confirm to me that you can see the PowerPoint slides. Is that okay?
Yes, now it's perfect. Thank you, sir.
Great. In fact, This is the first lecture I'm giving as as director of this new institute that has just been created, that takes my name, the Institute of Advanced Brain Studies, that is distributed across Brazil, the US, Europe, and Asia. Now we are about to make this announcement in a few weeks, but so this is the first after leaving Duke and coming back to Brazil. I decided to create a new structure that would allow us to disseminate all the knowledge and all the new potential therapies that I created over the 34 years or so that I was in the US. So it's a great pleasure to inaugurate this new institute with this talk. Well, I would like to show you that this field that today is so popular, particularly because different companies around the world are trying to do things that we already did now in human beings. And I will try to make at the end a balance of what is going on and why I feel that some of these initiatives are not going the right direction. But basically, I started my career in Philadelphia in '89 as a postdoc, trying to do with John Chapin, my advisor at the time, what you see here on the left side of this screen. This was the first time, a few years later, already at Duke, when we succeeded in recording for the first time in the history of neuroscience, 100 neurons, the extracellular activity of 100 individual neurons distributed over 4 to 5 different cortical fields in a behaving monkey. And what you see here is the activity. Each line represents the action potentials, extracellularly recorded action potentials produced by individual cortical neurons. And for the first time, we could actually see a sample of a whole circuit, the frontal parietal circuit, working in real time as this animal was preparing to make hand movements. And in fact, we could actually tell about 200 milliseconds ahead of time when the animal was about to make this movement, because we learned to identify the premotor activity distributed across all these areas of the cortex. And that was, as I'm going to show you in a second, that was the first shock. It was not only in primary motor cortex or the frontal supplementary motor fields that we could see motor-related activity. We could see this all over the parietal lobe and in a very highly distributed way. So that's the reason that the title of my talk is Brain-Machine Interfaces from Basic Science to Neuroprosthesis and Neurological Recovery, because it was this capability of recording large populations of neurons, individual neurons simultaneously, that opened our minds to create this new field and to try to link brains to machines. You're going to see why in a second. This is just a very brief summary. I'm not going to go through the entire slide, but it's a summary of everything we did in about 30 years of, since the moment we're able to develop technologies to implant thousands of microfilaments or microelectrodes. They are like hair-like, each one of them, in multiple areas of the brain, cortical and subcortical, to sample for long periods of time how these circuits operate in freely behaving animals. In fact, in Philadelphia, before I left, as a postdoc, John and I, we published a paper in Science for the first time showing the sampling of neurons across the entire trigeminal somatosensory pathway in rats, from the trigeminal ganglion to the somatosensory cortex in a freely behaving animal, and show how this whole pathway operated in real time as these rats were using their vibrissae, their facial whiskers, to actually sample tactile attributes of objects that we put in front of them. And that I think was a watershed event, because it proved to the community that a new type of neuroscience was starting. Because instead of monitoring just one neuron at a time, as we used to do for almost 100 years, we could now do hundreds of neurons simultaneously. And it was from this technology that I spent about 20 years studying neurocircuit dynamics and plasticity in behaving animals, rats and monkeys. And finally, from those studies, we derived some principles of population physiology, neuroensemble physiology, that led us to think that we had to design a new preparation, a new approach to try to understand how the brain encodes information, because it became pretty obvious to us, and I'm going to show you a couple of results before I go into the clinical aspect of this, why this was the case, that we should consider now populations of neurons and not a single neuron as the true functional unit of the nervous system. Well, I was also in Philadelphia that during a trip, I was already at Duke in 1997. I met John Chapin again, my advisor, my postdoctoral advisor, and we decided during a cheesesteak dinner that I think you feel very familiar with these circumstances at Gino's. John and I decided that to make the community understand what we meant by assigning populations of neurons as the true physiological unit of the brain, we had to be able to quantify the difference for a brain coding or for brain producing behaviors, of what would be able to get when we had a large population versus when you have individual neurons. And that's when we created this field. And this name was during that dinner that we came up with the name brain-machine interfaces to describe what we wanted to do. That was linking brains to a variety of robotic, electronic, and at the end, virtual devices so that we could test all the potential coding theories that were running around in neuroscience at the time, from individual neurons to populations of neurons. These are all, in terms of populations at that time in the mid-90s, all theoretical proposals. Nobody had been able to test it because there was no technology to record large populations of neurons. That's what we did. And it was from this decision in 1997, which I should say, during the dinner we were eating, and I don't remember who said, okay, let's call this brain-machine interfaces. And behind me, there was a huge truck driver who stood up, looked at us and said, that's a great name. So we got instantaneous peer review approval of the brain-machine interface label for this kind of research. Well, from here, we did a lot of studies in animals, and then we realized that there were potential, a huge variety of potential new therapies that we could apply to different neurological and more recently even psychiatric disorders. And that's how we got into, and I went back after, graduating from medical school, getting my PhD and never practicing. I went back to working with physicians and neurologists, neurosurgeons, trying to apply these discoveries in basic science to clinical applications. Just to show you the concept, this is what a brain-machine interface systems engineering diagram looks like. This is the original diagram that John and I published in Nature that opened the field wide open for many different applications. Of course, you have to sample the activity of the brain. It can be done with intracortical implants, with ECOG, or with non-invasive techniques like EEG. But then the whole thing needs to be, basically these signals that you're recording needs to be analyzed in real time. You need to use computer science and mathematics and signal processing techniques to extract from this sample of brain-derived signals motor commands that you can then, in less than 300 milliseconds, translate into digital commands that, let's say, a robotic device can understand. Because the central goal of this is to allow the subject to control the movements of this device just by producing the motor program in his brain that is required to produce this movement, let's say the movement of an arm or a leg. And instead of using its own, his or her own body, the subject will have to imagine this movement so that we can extract the motor commands that will be used to control a device that can be next to the subject. The subject can be wearing this robotic device like an exoskeleton, or as we demonstrated a couple of times, the robotic actuator can be in a different part of the planet, or it can be a virtual device in a computer screen. It's fundamental, though, that this device provides feedback to the subject, so the subject can judge how well it has been able to control this device just by producing brain activity that contains motor information. So this is the central strategy, the central experimental approach that we use in all the experiments I'm going to show you, and the clinical applications as well. To do the experiments in animals, we use invasive implants that we designed ourselves since the mid-90s, and we went through many generations at the Duke Center for Neuroengineering that I directed for 20 years. These are, as I said, hair-like microelectrodes made of tungsten or stainless steel microwires that are coated with an isolating material like Teflon or Isonel. So they're very flexible. When you implant them in the brain, they stay there for years, and you can keep recording for a long period of time. I'm going to show you in a moment. So you implant in cortical, subcortical structures, and you leave it in there. It's a single procedure. And once in there, you can start sampling things like this. This is a bird's eye view of one of the largest implants we did. This was about 2,000 microwires implanted in units of about 100 microwires, because I forgot to say, we can have two-dimensional arrays like this, 10 by 10. We can also have a high-density recording cube. It's a three-dimensional arrangement in which you continue to have 10 by 10, but then you have many recording contacts spinning off of this central dagger that constitutes the anchor of this z-axis that we created in this particular, we call it umbrella arrangement. So you can get up to 1,000 contacts coming out of these recording cubes. So this is a sample of about 1,874 neurons, individual neurons recorded from a single implant in a behaving rhesus monkey. We sample about 8 cortical fields simultaneously from the frontal to the parietal lobe. So we sample areas that are interconnected, very important for the generation of a variety of motor behaviors. And we use this animal for many years, as I'm going to show you, because this, besides the large yield, one of the great advantages of these implants is the fact that we can record from the same animals for close to a decade. In fact, in this paper that was in Nature Methods, here is the arrangement. It's a self-contained arrangement now, because we use wireless microchips that can broadcast these signals up to a few meters of a distance. So the animals are now untethered. They can actually, as you're going to see in a moment, freely behave. They can be behaving in a room or in a large enclosure, and we can be recording not only from one, but from several subjects simultaneously, because there are no cables anymore connecting these animals to any apparatus. And as I was saying, in this paper in Nature Methods, we follow 8 monkeys for more than a decade. And what we saw was that you can see here in this red line, this is a monkey that was implanted in mid-2009. We published this paper in 2014, but this monkey was recorded all the way to 2018. So we almost got a decade out of recordings. And here you see at different moments in time, the high quality of extracellular action potentials recorded from individual neurons, not necessarily the same over a decade, but the microwires remain alive for all this time because they don't damage the tissue. They are not rigid like these silicone daggers that some companies have produced and last just a few months in human subjects, for instance. In monkeys, the same thing. Our recordings are done with these flexible microwires that, once attached to the brain, move with the brain without damaging the tissue. And that's the reason we can keep it for many, many years, these recordings in both rats, monkeys, and more recently, people have shown that in humans. Well, let me show you the first brain-machine interface experiment so you can understand what we did later in humans. These are rhesus monkeys that were trained to use joysticks to control a computer cursor to cross a target that appears randomly on the screen. So every time the monkey crosses the target, it can get a juice reward. And monkeys love to do that, and they get very proficient, the 95, 99% correct, depending on the animal. And in this case, as the animal was learning to do this, we are recording simultaneously about 100 neurons, as you saw in the first slide, from 5 different cortical fields in the frontoparietal circuit, and sending these recordings, this data, to a wall of desktops at the time. They were running 21 different mathematical models to try to extract from this raw extracellular neuronal activity the kind of motor commands that the animal is producing to move its own arm, but we wanted to use that to train a robotic arm, a 7-degree of freedom robotic arm that was in a different room that we intended to use as the actuator when the brain-machine interface was trained and the animal could handle it. So when the animal became proficient in the task using the joystick, we play a trick on them. We remove the joystick, we turn on the brain-machine interface that was trained during the practice with the joystick, and now for the animal to move the cursor, it didn't have the joystick available anymore, so it didn't need to move its own arm. It had just to imagine which trajectories were needed for that cursor to cross the target. And if the animal could do that, it was because it was taking advantage of this brain-machine interface that was now on, because from that point on, the cursor's movement were controlled by the hand of the robotic device that was directly controlled by the animal's brain activity. So this is one of our pioneer monkeys, Aurora, doing this task. You can see here that she's controlling the joystick with the left hand, very fine movements, and she's trying to move the computer cursor, that little tiny white circle, into the big ball that appears randomly on the screen. She's very eager to drink the juice, so she's trying to cheat. And sometimes she succeeds, but she cannot do that all the time because there are 32 different random positions in which this target can appear on the screen. So she will have to really do the task to get the reward. And once these monkeys achieved about 95% correct, we play the trick on them. We remove the joystick, as you can see here. The joystick is gone. She's holding on the chair. And now we turn on the brain-machine interface. And the only way this monkey can now move this cursor into the target to get the orange juice that she craves is by imagining which hand trajectory or arm trajectory she needs to produce to get the cursor to go to the target, towards the target. And that can only be done now because as she imagines that, we are sampling about 100 neurons out of this circuit, and we are mixing their activity to make the robot reproduce her motor intention, and it's the hand of the robot now that is in a different room that controls the cursor. Oops. And as you can see, she goes there and continues to do the task. Even the cheating is reproduced by our 21 mathematical models. This is a two-dimensional task, but a few months after this experiment was done and published, we did a three-dimensional task, and we included force, gripping force. The animal now had not only to move in 3D, but it had to produce a certain amount of force to grip the target. And she was able to do that, as you are seeing here. All these trajectories are produced by the brain-machine interface controlling a hand that is dealing with the movements of the cursor and getting her to drink the juice. When we analyze the data coming out of the multiple, the 5 cortical areas that we're recording this experiment, we are pretty shocked because nobody had reported this before. These are neurons in the x-axis, the mass of neurons recorded simultaneously. This is how much they contributed to a given model, a given linear model to produce, let's say, hand position in real time or gripping force. And each color represents a cortical field. The navy blue is the primary motor cortex. Dark red is the primary somatosensory cortex. Premotor dorsal cortex is this purple here. I'm sorry. And the green is the posterior parietal cortex. And we had different amounts of neurons recorded in these areas, but what you can see is that there is information about both hand position and gripping force all over these cortical fields. A primary motor cortex is still the best. You know, if you have 15 neurons, for instance, recorded simultaneously there, you get the best contribution to the models. But depending on which parameter you are trying to reproduce, let's say, gripping force, the posterior part of corticals for 30 neurons is pretty similar to the primary motor cortex. But the key message here is the coding for this motor behavior is widely distributed all over the cortex. Many years later, we put electrodes in all sorts of cortical fields, and we could still recover information about moving the hand, not only in the motor cortex. The other major message is that if you start removing neurons from this population that you're sampling, by the time you get below 20 or 10, depending on the cortical field, your performance goes down very quickly. So when you have just one individual neuron, no matter how good this neuron is, your performance is dismal, and the monkey cannot sustain the control of the cursor as you saw. So single neurons are not the way the brain operates. Despite everything that you read in Kendall and Schwartz, we have to rewrite all these textbooks because it became obvious with 10 years of these experiments that the functional unit of central nervous system is a population of neurons, not a single neuron. So Donald Hebb was right all along. He said that, and nobody believed him in 1949. And it took almost 50 years exactly 50 years for this to be demonstrated experimentally in a categorical way. So I'm just going to show you a couple of variations because then you understand how we translated all this to clinical practice. This is what we call a brain-machine-brain interface. We published this in 2011, initially in a paper in Nature and later in 2019. And in 2022, a group in Pittsburgh in one patient has basically applied this concept to this quadriplegic patient. Unfortunately, I forgot to bring that movie that they published, but the idea was there a decade before in monkeys. So first, we created a brain-machine interface in which, as you saw, a monkey can control a virtual arm now. This virtual arm can scan the texture, the surface of spheres that visually look identical. You cannot differentiate them just by looking at them. Like a monkey could look at them and not know the difference between them because the difference is in the microtexture of these objects. And you can only detect the microtexture, the different microtexture, by scanning your hand on the surface of these objects. And if the animal does that, controlling this virtual arm, the virtual arm is going to send directly to the primary somatosensory cortex electrical pulses that are proportional to the microtexture of these objects. And there's one of them that has a very fine microtexture, about 200 hertz. And that's the one that the animal has to indicate to us that he has detected, because if he presses the center of the texture, he's going to get juice. So this is a brain-machine-brain interface because the body of the animal is never involved in the task. It is controlling the virtual arm to scan the object and is receiving feedback directly to make a tactile discrimination decision, which object I should press to get the reward. And that's exactly what they do. What you're going to listen here in the background of this video clip is the single neuron from the primary somatosensory cortex firing while the animal is scanning the objects and making the correct decision. Of course, for you, I have added labels to show which one is the correct target. The monkey didn't have the sound and didn't have any labels to help in the discrimination, and the objects are changing position every trial. So the animal literally has to use the virtual hand commanded by its own brain to detect the microtexture by basically analyzing a pattern of cortical microstimulation that we deliver to the animal, to the animal's brain directly. So this is the animal controlling this avatar of its hand. And when it finds the correct target, it presses on the center. And that's the reason you hear the beep. The beep is the solenoid delivering the reward to the animal. So as you can see, it has to scan the surface before it makes the decision. And sometimes it's difficult because the textures are also very similar. But nonetheless, the animal learns to do this task as if it were using its own fingertips. And it gets very, very good at it. So he can drink about 500 ml of juice just by doing this test. So things evolved very quickly from that point on. And in a few years, it took us 5 years to publish this paper. But in 2009, we start playing with the idea first of using a wireless interface so that animals now could learn to control devices that didn't look at all like an arm or a leg or any piece of the body of these animals. In this case, for instance, we taught these animals to use a brain-machine interface now with 300 neurons being recorded simultaneously from their cortex to drive an electronic wheelchair. So this was the first self-driven car controlled by brain activity alone. And let me tell you, one day before the pandemic in 2019, actually a couple of weeks before the pandemic started, I was in Bonn, in Germany, giving this talk and presenting this specific experiment to a bunch of executives, CEOs and VPs for research of the automobile industry, organized by Daimler Mercedes. And I said, you know, you guys are talking about self-driven car that actually don't work. Let me show you how a monkey does that using a brain-machine interface. And you can imagine how shocked they were and how mad they were that here we are in a little lab in North Carolina doing something that they dream about and so far have not been able to accomplish very well without a brain-machine interface, of course. So what you're going to see now is how a monkey that is placed randomly in 1 out of 3 positions in the corner of a room is able to, from trial to trial, generate a new trajectory that will allow this monkey to drive to the other corner of the room. It could be a hall. We did that in the major hall. I don't have that movie here, but to collect a reward just by using this wireless brain-machine interface. First, you're going to see images from a camera on the ceiling. So you see now the monkey driving, collecting the grapes. We put it back in a different position. Now it has to improvise a new trajectory because it is only using the brain activity to control the true electrical motors of this wheelchair, and it has to get from point A to point B by itself. Here is from the front. You see that the monkey has realized that there is a free lunch in America. You just need to have a brain-machine interface working. So it doesn't need to move. You just need to collect the grapes and enjoy it. And as I said, the monkeys learn to do that in a very proficient way. And we have a shock result when we analyze the neurophysiological data that we collected in primary motor cortex, in premotor dorsal, in the primary somatosensory cortex. To our shock, when we did the analysis that people that work in the hippocampus usually does, so we plotted the two-dimensional space of the room. This is the area getting close to the pod where the animals collect grapes, and this is where they come from. What you see in yellow here is a tuning curve for space. And you can see that neuron 1 on top and neuron 2 on the bottom. Of course, we had thousands of these neurons. I'm just showing 2 examples. They were tuned like place cells are tuned in the hippocampus for extra personal space. But this is not the hippocampus. This is the primary motor cortex. And if I would put neurons in the primary somatosensory cortex, you would see the same thing. And you, as me, as I have learned that place cells were, you know, the landmark finding of the hippocampus. In fact, a good friend of mine, Dr. O'Keefe in England, John O'Keefe, was able to get a Nobel Prize with his students for this amazing finding that he was able to observe in the late 70s. Nobody believed him at the time. It took him almost 40 years to convince the entire community. And I was giving this talk a couple of years ago at UC London, and John was there in the audience, and he was very happy and very surprised because we were finding his beloved place cells in the primary motor and somatosensory cortex, very low in the so-called cortical hierarchy, very far from the hippocampus. So this is there, and it shows you that our notions of cortical hierarchy may have to be revised pretty soon because it doesn't fit anymore, the experimental data when you have freely behaving animals. This was very important because we could combine these place fields to reproduce the trajectories that these animals created with the brain-machine interface, like people do in the hippocampus. And it was a very similar result. We could linearly reproduce by putting together 300 neurons simultaneously recorded the trajectories that these animals were doing with our brain-machine interface. Well, back in 2002, when we first published our data in monkeys, John Chapin and I published this cartoon in Scientific American. This is 20 years ago, showing that, or proposing at the time, that if a paraplegic patient had a lesion that blocked motor commands coming from the cortex to be delivered to the periphery of the body, and of course, making them paraplegic, and not only paraplegic or quadriplegic, not being able to move, but also not able to receive any tactile information coming up to the brain, so they could not feel their bodies at all. What we proposed then is that our findings suggested that in the future, patients with paraplegia or quadriplegia may be able, and other patients, other neurological patients, may be able to take advantage of what we had just published and use a brain-machine interface to record the motor intention that they were still capable of producing in their cortices, and through a computational mathematical bypass to the lesion, control robotic devices, including what at that point we call a robotic vest, a new body that these patients would learn to control. And because these bodies could send feedback back to the subject's brain, they would be able to regain mobility by using this device. Of course, at that time, people thought that we are two just crazy lunatics coming from Philadelphia who had lost their marbles thinking too hard about their own results. It turned out that this proved to be true, and in very peculiar circumstances. About 10 years after we published that, I was here visiting Brazil and had the chance to talk to the Brazilian president at the time, and talk her into the idea, the crazy idea, because she was not as conservative as peer review is, to actually, since Brazil had been awarded the World Cup, the Soccer World Cup in 2014, what would she think if we could do a demonstration in the soccer field of a paraplegic using a non-invasive, now no implants, a non-invasive brain-machine interface to control a lower limb robotic exoskeleton that could allow this paraplegic patient to stand up and kick the ball and, deliver the kickoff of the opening ceremony of the World Cup here in Brazil, in a soccer field a few miles away from where I am right now. And she looked at me and said, that sounds a pretty good idea if you can do it. I said, well, I think we can do it. We have done lots of experiments in monkeys and we are ready for this. Besides, I have the help of about 156 people who are willing to drop everything they're doing in life, open their patents, drop their labs, and come to Brazil for 6 months to actually prepare for that. And long and behold, the Brazilian president looked at me and said, okay, let's do it. And she just signed a presidential order that allowed Brazil to fund that, because I tried to do that in the United States, but NIH would not have it at that time. That's pretty surprising now, but at that time, even though 2 years before, I had received the Pioneer and Transformative Awards from the director of NIH, they thought it was too much to try to do that in humans in the Super Bowl or something like that. Well, I chose something a little bigger, the World Cup. And here we went. We basically built from scratch a state-of-the-art neuro rehab facility here in São Paulo. And then we recruited patients and an international team, as I showed to you. These are some of our patients who were chronic paraplegics from the Brazilian National Database, which at that time had about 65,000 people enrolled in the program in the largest spinal cord neuro rehab hospital here in Brazil, in São Paulo. And basically, this is the newest facility that we now have, is a 5,000 square feet, 500 square meter facility in the northeast of Brazil, a state-of-the-art facility that develops new brain-machine interfaces and new applications that I'm going to show you in a moment. But these were the original 8 patients. They're all Asia A or Asia B complete paraplegics, because different from some recent demonstrations, we didn't take incomplete spinal cord injury patients that already had voluntary motor control of important muscles in their lower limbs. We took people that were for 6 to 13 years, 5 to 13 years, totally paraplegic, no recovery whatsoever, Asia A's or Asia B, repeatedly confirmed by different groups of physicians, including our own international team. And then we decided to incorporate everything that we have done in monkeys in a non-invasive way, using now multi-channel electroencephalography to, first of all, train these paraplegic animals to incorporate a new body. By using virtual reality, we use a brain-machine interface that allows these patients first to control the movements of an avatar soccer player in a soccer field. And every time, as you're going to see in a moment, every time this soccer player touched the ground with its virtual feet, the patient receives a tactile feedback through a haptic display on the surface of their forearm skin. And that allowed them to have a very realistic visual tactile feedback of an avatar walking under the control of their own brains. As the patients became proficient in this task, we moved them to different tasks in a sequential way, up to the point where they start using off-the-shelf robotic devices, robotic walkers that they could control with the EEG activity. And finally, after a few months, we moved them to a custom design exoskeleton, a lower limb robotic device like we had mentioned in 2002, that could be controlled directly by the EEG, and that was instrumented with sensors that I'm going to show you in a second to provide tactile feedback to the subject as it learned again to walk again using this device. This is the first version of that exoskeleton. We are now in the 3rd generation of this machine. I'm going to show you in a moment the 3rd generation, but this was the original pioneer exoskeleton. It's an electrical hydraulic device controlled by brain activity, and is a hydraulic because the person triggers the electrical motors, as you saw the monkeys doing with the wheelchair, but these commands go through analog lines, hydraulic lines, to control the motors all over the leg, because the patients wanted a smooth movement, not a fractionated robotic-like movement. They wanted to show themselves to the world as humans, not as robotic devices. And we spend a lot of time to fulfill their dreams in that domain. This is how the exos were instrumented. We printed circuit boards that had pressure and distance sensors deployed all over the feet of the exo, so that when the patient touched the ground, a pressure signal would be generated that could be delivered to the sleeves that they were using on the forearm, so that they could feel the ground, the texture, and the characteristics of where they were walking on. So this is, for one of these 8 patients, the first day. Everyone, of course, went through this, but this is the first day one of our patients, Asia A, completely paralyzed, used this device for the first time to walk after many years in a wheelchair. And you're going to see that he's looking straight ahead because there's a huge mirror to provide visual feedback to the patient as he walks for the first time, using this brain-machine interface to control the exoskeleton movements. Oops. What happened here? Oh. So you see the go-ahead lights that tell the patient that he can start. And now he's walking and receiving this feedback on his arms, forearms, and he's looking at himself walking upright. Later on, we published a study showing that this visual tactile feedback was essential for the patients to incorporate the exo as an extension of their body schema. So the cortex literally incorporated the exoskeleton as an extension of the patient's body, And we did a lot of psychophysical studies to demonstrate it. So you see that the patient was regaining mobility. That was our main milestone. And in fact, I cannot show you the opening kickoff of the World Cup because FIFA never relinquished the copyrights to us. They wanted us to pay a couple million euros for the copyrights. And of course, scientists don't have that kind of budget. But before we went to the soccer field here on June 12, 2014, we did a final test with Juliano Pinto. This is the guy who did this kick. He was a T4, complete Asia A, paraplegic, no movement of the legs, no sensation of the lower parts of the body for, at that point, 10 years. And what you're going to see is what happened on the soccer field. We put these LEDs all over the exo to illustrate to the audience. There were about 65,000 people in that stadium and about 1.2 billion people watching that over the internet and TV. And when you see the LEDs flashing, you will see that it means that Juliano is ready to control the exo, and he's going to imagine kicking the ball, and those controls, those motor commands are going to be translated so that when my student put the ball in front of the leg, Juliano could kick it. And that's exactly what happened on the pitch, on the soccer field. So Juliano is now turning on the exo. He's ready. The ball is placed in front of him, and he was able to kick the ball and to receive tactile feedback to feel the impact. And for someone who was for a decade confined to a wheelchair, that was quite an experience that moved, you know, the entire stadium, ourselves, and this audience all over the world. But even though everybody, including ourselves, thought that this was the end of the story, it was not, because a month later, we came back to the lab, and we were doing our neurological examination, a routine required by our IRB here in Brazil, and we had not seen anything changing, and the patients never saw any type of recovery for all the time that they were diagnosed with the spinal cord injury. However, 1 month after the World Cup, we brought Juliano, the guy you just saw. He was, as I said, a T4 originally, but when we did our neurological examination by this international team of neurologists, Juliano suddenly was classified as a T11. He had regained some outer type of tactile, not a totally normal, but some sort of outer tactile capability, 7 dermatomes below the level of the original lesion. We could not make sense of that, so we started bringing all the patients as we were required to do, and suddenly we saw that everyone was having some sort of tactile recovery below the level of the injury. In fact, at that moment, the average gain in outer tactile sensation was 5 dermatomes below the level of the injury. Well, we followed these guys for 28 months, and as you can see here in the blue line, as time went by, this tactile recovery, because they continue, 7 of the 8 continue to train with us for 28 months, average reached 10 dermatomes below the level of the injury. Juliano, for instance, who was originally a T4, at 28 months, he was feeling his toes. And he could identify which toe we were, with a pin touching when he was blindfolded. So he had some sort of recovery, tactile recovery. Well, that was not all. A few months after we noticed this improvement, we start noticing something totally unexpected. So we use a system to suspend these patients since the beginning of the protocol, and we always ask them to try to walk on the air if they could. And of course, they never did any movement, voluntary movement. But one day, about a year into the training, although we had noticed things with 8 months before, but this is the unprecedented result we got. We asked this patient to do the same thing, and suddenly she was moving her right leg. She was flexing the hip, flexing the knee, and extending the ankle, and trying to do some steps on the air. And that was totally shocking to us. So we, of course, could not believe in this. We kept quiet and kept monitoring all these patients. A year later, the same patient was doing the same kind of task, but now we are recording EMGs all over her body. and doing all sorts of measurements, and she did this on us. When we asked her to walk in the air again, as she did a year before, she first did what you saw before. She started moving her right leg, and suddenly she started doing the left leg. So she basically was experiencing a recovery of voluntary motor control of several muscle groups below the level of her lesion, what originally was T11. She was also Asia A originally, but she upgraded to Asia C during the training. And in fact, when we look at all the 7 patients that remain with us after 12 months, one patient had to drop out at 12 months because he moved out of São Paulo. What you can see there is that during this training that combined non-invasive brain-machine interface, virtual reality and robotics for the first time, patients start being upgraded, and all 7 at 28 months were classified or reclassified as partial paraplegics. They were now Asia C instead of Asia A or Asia B. I don't have time to show all the results, but all patients regained a degree of voluntary motor contraction. They all had significant increases in tactile, altered tactile sensation, including proprioception. And they all had significant improvements in bladder control and bowel movement control or production. So it was for someone like me who was taught in medical school in the 80s that I would never see a spinal cord injury patient recover from a complete spinal cord lesion. This was the shock of my life from a clinical point of view. We did a second group of patients later. We reproduced this result, and we just learned that a group in China using our similar protocol has just reproduced our results with a large control group that we didn't have in this first study. We had in our second study, but it was a small control group. Now they had a very large control group, and they reproduced the motor results identical to what we got here. In fact, the improvement of these patients was so tremendous. In some of them, in 3 of them, we are able to create a new approach in which after their being upgraded to Asia C, we use non-invasive functional electrical stimulation controlled by EEG to actually allow these patients to walk without any exoskeleton. This is one of these patients that was originally Asia A. He was upgraded to Asia C during the training. And as you can see here, with a simple $10 walker and a little bit of body suspension, he was able to basically regain autonomous gait or autonomous walking by combining whatever he gained with the recovery with our protocol with this multi-channel brain control functional electrical stimulation of the legs. In fact, we just realized that the results we obtain with the non-invasive walk again protocol in terms of motor improvements and sensory improvements, but particularly the motor improvements, the index, the whiskey that we use is absolutely identical to all the studies that have tried to demonstrate the same kind of thing with implants in the brain. There was a paper that came last week with a single patient from the Cochin group in Geneva, in Switzerland. Actually, that's the Center of Neuroprosthetic Devices that I created during my sabbatical in 2005 and 6. I created that institute in Switzerland, and then, of course, I returned to Duke. Well, that group just published a study that made news all over the world with ECOG controlling a spinal epidural spine implant to make people walk, one patient walk. And they published their WISC index. And we went to look at, and after 48 months, that's what it took for that patient to be able to walk with that ECOG and spinal implant. He has 40 months of training with the spinal cord stimulation, and then the implant at 40 months and 8 months of training with the ECOG. Well, the whiskey is identical to ours, and we got that with 7 to 12 months, depending on which patient you're talking about. And now we have a sample of more than 30 patients. So you don't need, as you have heard in the news, or you have heard Mr. Elon Musk saying that you need a cortical implant to make paraplegics walk. No, that's not true. You can do it with non-invasive brain-machine interfaces, and you can spare your patients a neurosurgical procedure and a spinal implant for this type of pathology, Asia A. And by the way, the patient from last week was an Asia C all along. He had a voluntary motor contraction on all major muscles in the leg. They had what we call fours in terms of muscle control index in all major muscles, while the patients you saw here had 0 in all muscles when we started. So you don't need an implant for that. You can do it in a non-invasive way. However, just to finish, this is our third generation of the Exo. Is a non-invasive EEG control, but now instead of the backpack that you saw that was worth about 30 kilos of electronics, the entire Exo is controlled by an iPhone that the patient can wear on the sleeve. So there is no issue on that. If you allow me 3 more minutes, I would show you that there are events where implants will be very useful in the brain-machine interface field. And this is an example. In 2009, we discovered that if you stimulate the spinal cord with an epidural implant in rats and later in monkeys, animal models of Parkinsonian gait freezing, we were able to make these animals move again because we actually treated the gait freezing neurophysiological correlates that we found in the brains of all these animals and more recently in humans as a seizure, as an epileptic seizure. And we use high-frequency stimulation of the dorsal column of the spinal cord as literally as a defibrillating microchip. And what we notice is that if you do that, the seizures that are happening when the animal is frozen because it's depleted of 99% of dopamine in these transgenic mice, the animal moves again. We published this on the cover of Science in 2009. This is the original result. This is a mouse, a transgenic mouse that was depleted of 99% of dopamine, so it's frozen. But when we start the stimulation of the dorsal column of the spinal cord at 150 hertz, this guy suddenly starts walking, and he goes all the way to the other side of the cage to collect what he craves, which is sugar water. So the animal, in 2 seconds, now we start the stimulation at 150 hertz, and the little guy says, okay, now I can move, because basically the the beta seizures that we identify in the motor system of this animal now have been defibrillated. They are out of phase now. The neurons are now able to fire out of sync, and this animal is able to actually move. Well, people here in São Paulo and in many places here at the University of São Paulo, a few years later, they decided to test that in gait freezing patients that already had received DBS and didn't work, of course, deep brain stimulation, as you know well. And they implanted spinal cord stimulators that are used for chronic pain at the cervical thoracic location that we use in our experimental animals. And then they did exactly the same protocol that we did. And they published the first 4 patients. There are now more than 100 patients published. These are Parkinsonian patients that have already, as I said, received deep brain stimulation, but the gait of freezing could not be treated, as you know well. On the left is the patient without spinal cord stimulation. On the right, the spinal cord stimulation is on at the same time the DBS is on. And you can see immediately the difference in terms of walking for these patients. On the left, again, the patient has only DBS. On the right, DBS and spinal cord stimulation. This is the 20-meter task. The patient now doesn't need the assistance of the nurse to walk, and it has balance. It is not at risk of falling. And you can see at the left how difficult is that for a patient, the baby steps to walk. You are going to see a couple more patients, 3 more patients, and the second patient is pretty amazing. In fact, I told him that he could go and play soccer already, because the result in terms of gait improvement was tremendous, but primarily balance control too for that patient. That's patient 2. Again, on the left, you see DBS on, but spinal cord off. On the right, the spinal cord stimulation of the cervical thoracic region of the dorsal columns is on. And the other 2 patients, what is very interesting about them is that the cardinal symptoms of Parkinson's, which they had in a very severe way, tremor, rigidity of face, expression, they also got improved by the spinal cord stimulation, something that we didn't predict originally in our rats and monkey studies, because it's very difficult to measure that clinically in these animals. But in humans, several groups now are reporting that these implants also have a significant effect on the cardinal symptoms of Parkinson, in addition to the gait of freezing being relieved in a significant way. And some groups have already done that with brain-machine interface controlling the stimulator. And recently, a group in Japan had taken one of our ideas that we didn't have time to test in humans. Okay, this is the 3rd patient now. You can see the difference in gait, but also in the cardinal symptoms of Parkinson's. But this group in Japan has now done a control and experimental group with 50 patients in each group, but using non-invasive TMS stimulation of the same region of the spinal cord. And they got results that are very similar to what you're seeing here with the stimulator. So this is just to show you that this is just the beginning of a complete new, in my opinion, complete new age of interactions or therapeutical approaches to neurological disorders. This is just to show you that recently we published another paper in chronic epileptic models in animals, in which the same approach of dorsal column stimulation of the spinal cord was able to, in a 70 to 8% of the episodes of seizure, PTZ-induced seizures, we are able to desynchronize and block the seizure activity by using the same approach I just described to you. So I actually believe that we have just touched the tip of the iceberg on what we will be able to do. And the great news is that a lot of that will be done with non-invasive approaches. And just to, I don't have time to show these shared brain-machine interface approaches. I'd like just to thank some key people that participate in these studies at Duke for 28 years. They are here on the left: Michael Lebedev, Eric Thompson, Gary Lee, you are a technician. Here in Brazil, 2 of the main investigators. Suleiman now is a professor at the EPFL in Lausanne, just published a very beautiful science paper a couple weeks ago. Gordon Schenk at the Technical University of Munich, and Hannes Blorer, who was a great friend of mine at the EPFL, now retired, who did some development with us. Thank you very much. If anyone has questions, I, of course, delighted to answer.
Yeah, Dr. Nicola, I just wanted to thank you for this fabulous talk. Any questions from the audience? Yeah.
Hi, good morning, Dr. Nicolelis. That was an informative talk and fantastic. I just had a quick question. If there was a significant difference, perhaps, in the efficacy and mobility of these patients based on the level of muscle atrophy and also, like, perhaps spasticity that you saw among the patients?
That's a very good question. As you know, we had patients with more than a decade of spinal cord lesion, and they had tremendous degree of muscle atrophy. Our inclusive criteria also try to include in these studies people with low levels of spasticity to start with. So we did, I didn't have time to show, but in the papers that we published, we show how much the training improved the muscle atrophy, because we are continuously measuring this progress over these 28 months. And when we combine everything you saw with the FAST, there was also a tremendous improvement, a significant improvement in the ability, in the atrophy levels of these patients. So we did pay attention to that. And our philosophy was what we call share control. Because our patients, all the patients we interact with over the years told us, we don't want to be robots. We don't want to be left inside of a robot and, you know, the robot carries us, you know, passively. We want to be active players. And I believe that what we did with them of allowing them to be active players of this therapeutical approach, including the ability to produce voluntary motor brain activity that drives the entire device, was essential for the kind of therapeutical approach and for the recovery, the partial recovery that they experienced. Because in a group of patients that we did with no active control, just passive movements of the exo, there was no improvement. So no neurological improvement. So you need to make them participate voluntarily, and they have to be highly motivated. And the psychological analysis that we did for 28 months shows that clearly. Most of these patients were borderline depressed when we got to them, okay? And at the end of the process, they got jobs, they got out of the house, they were able to go and, you know, interact with the world out there, and the psychological profile of these patients changed dramatically. So this was also a secondary result that we didn't expect, but it was very important to see how the quality of life and the well-being of the patients was improved by the fact that they were able to regain autonomy.
Thank you.
Thank you so much, sir, for your time. And again, if we have any questions, we'll reach out to you. I have your email. So thank you so much.
I appreciate it. Thank you. Have a good one there.
All right.
Bye-bye.